Jason Park, the CEO of Empress Therapeutics, joins us in the studio this morning at Flagship Studios in San Francisco. Jason, thanks for coming back to the studio today.
It's a pleasure, Lindy.
Great. So, Jason, I have heard you describe Empress as the Google Translate of genetics to chemistry. Tell me a little bit more about what that means.
Well, to me, one of the domains where AI has really been shown to work quite well is in the interpretation and the manipulation of language and code. And one of the basic facts is the most important molecules to human life are encoded in the code of life, DNA. DNA encodes RNA, encodes proteins. Empress is one of the first companies to really extend that to the world of chemistry. And so you can sort of think of us as the Google Translate or the Empress Translate of genes equals chemistry.
Neat. And you're doing that using metagenomics, which is a little bit different than traditional genetics drug discovery. Tell us about why metagenomics and how that gives you an advantage.
So one of the things about small molecule drug discovery is, if you think about the challenges, one of the challenges is the search space is effectively infinite. It is just so large. And what we've realized is inside the human body is an entire universe of chemistry that interacts with every possible drug target that's already inside of us. And a lot of that chemistry actually already looks drug-like, has drug-like properties. And a lot of that chemistry is actually made by microbes that are in and inside of us. So microbes are really good chemists, and you have all these medicinal chemists that have been inside of us since the dawn of time. And what we've been able to figure out is that because they're synthesized by microbes, that means there's actually a genetic basis. You can look for the genes that encode those chemistries inside the metagenome. And so that is what the metagenome is. It is the entirety of all the genes inside the human body. And it turns out there's actually 170 million genes inside the human body. And we've been able to find drug-like compounds from that.
And that's where you're taking the translation piece from those genes into chemistry.
Absolutely, yeah. And to your question, why is that an advantage or why is that different? One of the challenges in small molecule drug discovery are threefold. You have to have a good target. Even if you have a good target, you have to have the right indication. And then you have to overcome toxicity. It's traditionally been a problem. So by looking at chemistry made by microbes that act on human disease targets, we start first with human data. We look at patients. We compare them to healthy individuals. We figure out which compounds are important on a genetic basis. So we know the indication from the start. We know the targets are good because evolution selected them, and we start with compounds that have some basis for thinking about their safety because they've already been inside the human body. So we're overcoming the challenges from the start. So that's what our advantage comes from.
So that sounds like it's giving you a head start in a lot of different ways, both in knowledge and also in time. And as you've advanced Empress over the last year, it sounds like you're starting to bear that out. Can you tell us a little bit about what you have accomplished in the last year, and in just 2 years, shortening that time frame from start to a drug candidate, for example?
So I'll start with that last point, actually. So with one of our first programs, we went from program initiation, just looking at diseased individuals, comparing them to healthy, to a development candidate in under 2 years. So we think that's about a half to a quarter of published industry stats on speed. So we have multiple programs, actually. We have 3 first-in-class programs. And again, back to that concept of the basis of, if you start with genetics, you start with patient data, and you start with compounds that have some basis for thinking about their safety, that really should read on future success. And we are seeing that in the preclinical setting.
So talk to me a little bit about, you mentioned 3 programs starting to progress towards IND-enabling studies. Tell us a little bit about how that pipeline is shaping up and what timelines you're looking at.
So we have 3 first-in-class programs across 3 different autoimmune immunology indications. One of those has already been partnered out. We've applied our platform to multiple broad disease areas. I mean, we're talking about chemistry inside the body against every possible disease target. So we've looked at immunology, we've looked at oncology, we've looked at metabolic disease, we've even looked at neuro and infectious disease. We have hundreds of potential programs. We have hundreds of compounds that have a genetic basis, that have a genetic link to different diseases, that have a human relevant component built into them. So as we continue to flesh that out, actually partnering is going to be a big part of that.
Well, tell me a little bit more about that partnering strategy. both on the pharma and the drug discovery side, but I imagine you talked a little bit about Google Translate. There's got to be a tech component to that too.
Yes, so there are several companies that we're speaking to that have helped us develop some of those proprietary technologies. There are partnerships where we've acquired patient data and generated proprietary data sets from clinical samples. And then really at the end of the day, though, it's about making medicines for patients. And we're a small team. We're still under 50 people, and we have hundreds of potential programs. So obviously working with partners is going to be critical to getting medicines to patients faster.
So, Jason, as you've progressed your programs this year, I've heard you say you are seeing 50% success rate in vivo, very clean toxicity profiles. What do you attribute that success to?
Well, it goes back to that starting idea of what are the challenges when you're trying to discover and develop a small molecule drug. And historically, it's been, do you have a good target? Are you developing it in the right indication? And can you overcome tox? And for us, again, by starting with patient data, comparing individuals with a specific clinical phenotype or disease to healthy individuals, and using genetics to figure out what programs to work on, that is translating to good targets, and we know the indication from the start. And by working with compounds, again, that are made by microbes, synthesized by, actually encoded in their genes and synthesized inside the human body, there's a reason to believe in inherent safety, or at least compatibility with humans. So that ultimately will translate to clinical success. And as you pointed out, we are seeing this in the preclinical setting. When we've gone into disease models, informed by the patients, we're seeing 50% of the time bioactive compounds with exceptional clean and tox profiles, clean safety profiles and tox profiles. So that gives us a lot of confidence that what we're seeing is going to translate.
It's very exciting, Jason. It sounds like you truly are translating ideas into potential products. Sounds like an exciting year ahead.
Absolutely. We're looking forward to it.
Great. Thanks, Jason.